in the next 15 minutes or so, instead of overwhelming you with data, what I would like to do, I will tell you a short story about one of our current research interests in my lab in relation to obesity-linked different types of cancer. Although I will focus today on obesity-linked hepatocellular carcinoma, but the factor which is linking this disease process is common to different types of cancer. So let me start with the burden of liver cancer worldwide. This is the latest data which is available from the IARC. In 2012, almost 800 new cases were diagnosed of liver cancer worldwide. This liver cancer ranks 5th in men and 9th in women. So this gives you a sense of there's a sex differences in the incidence of liver cancer. Almost similar number of deaths were also reported during the same period. However, it ranks 2nd in death from cancer worldwide. So what this means that we have a very poor grip on this cancer. So mortality to incidence ratio of this cancer is very close to 1. And what is most importantly, this is geographically similar pattern throughout the world, which is clear from this graph, which is taken from the same sources. This graph basically shows the incidence and mortality of liver cancer in the different parts of the world. Blue bar represent the incidence and red one represent the mortality. And as you can see that, although the incidence vary differently in the different parts, but the mortality goes parallel with that one throughout the country. So it really doesn't matter which part of the world you live in. If it's a strike with this cancer, the chances are very low. In Canada, this is a crisis in the making. And this is a report of the burden of this disease by the Canadian Liver Foundation, which is published in 2013. Coincidentally, the same year, liver cancer was a special topic in the Canadian Cancer Statistics, which is published by the Public Health Agency of Canada. I will show you a couple of graphs from this report. Although this is Canadian context, but this is also applicable to different parts of the world, including the United States. So this slide shows the incidence of SCC in Canada is spanning a period of almost 3 decades. And as you can see that throughout the 3 decades, basically there is an increase in the incidence of SCC. And as you can see, there is also sex differences, which is clear throughout the year. If you look at the mortality, during the period like 2000 to 2007, and upper line basically represent the SCC. SCC is the most predominant form of liver cancer, which accounts for more than 75% of liver cancer. So if you look at the number, there's a more than 50% jump during a short period of 8 years only. So this is a stunning increase in mortality during a very short period of 8 years. Even most worrying is the fact that there is no any sign of coming down. So it's continuously going to be rise. This is the projected incidence of SCC, and mortality is going parallel with this one. So there is no chance. It's plotted up to 2020. You can see that. So this continuous increase, and liver cancer is the only cancer in Canada whose incidence and mortality both are continuously on rise. And one of the unique thing about liver cancer is that liver cancer is very different in one way from the other cancer, because liver cancer is always associated with the underlying causes like hepatitis B or C, diabetes, obesity, or alcoholism. Okay, so there is a strong evidence, epidemiological evidence from the literature showing that the obesity and diabetes basically increases the liver cancer 4 to 5-fold. And this is the American study of 900,000 cohort. Similar report from the European study showing that with the increase in the BMI, there increases the risk for the SCC or liver cancer by 3 to 4-fold. This is another analysis of the CR Medicare database in the US. Almost 18,000 cases of SCC. Out of these 18,000, 60%, close to 3,000, were the biopsy-proven fatty liver without any sign of any other etiology of SCC or liver cancer. And out of these 16%, 36% were non-cirrhotic, and 18% were without any sign of NASH. Because historically it has been always associated with the liver cancer with the cirrhosis or the NASH. However, these data suggest that the non-alcoholic fatty liver disease without cirrhosis is an emergent and independent risk factor for the hepatocellular carcinoma. Okay, but there's a strong epidemiological, but we don't have a very good understanding of the mechanism of how this obesity leads to the different SCC. That there are a number of challenges and limitations in the field. The first challenge is that currently there is no animal model available which is spontaneously developed obesity-linked SCC in a progressive manner. So to circumvent that, what people try to do basically combine the two different models to make a model of obesity-linked SCC. And to do that, generally the obese model used is genetically obese, either OB, DB, or diet-induced rodent model, and combining them with the transgenic or carcinogen-induced model. The problem with this approach is that you are talking about obesity, and when you combine obesity, it's always, there are a number of factors which coexist together. So the problem is discerning which one is the driver or which one is just the bystander in this process. When you use the carcinogen-induced model, so the mechanism involved in carcinogen-induced model may not necessarily be the same, which happen in the case of human, which is it develops spontaneously in relation to the systemic changes in the body. The other problem is that you cannot do this mechanistic study with the human study, at least in the case of onset, because this cancer is always diagnosed late diagnosis because there is no symptom at the early stage, and the long follow-up that will require to do the mechanistic study. So the obesity-linked cancer is a very complex disorder. If you look into the literature, all factors have been implicated in connecting the obesity to the cancer development. But the problem is this all coexist together when you talk about the metabolic regulation. So the real challenge is what are the real driver in this one? Okay. And this is further complicated by the fact that the SCC is sexually dimorphic. You have seen already in previous few slides. Okay, we know that it's related with the sex steroid, but what we don't know is basically mechanism, how the sex steroid basically leads to sex differences in SCC incidence. The focus has been always on the hepatocytes and liver, but we don't know whether other cell type and tissue types are also involved in the sex differences in SCC incidence. And I will give you some data on that one. So the question is, what do we need, how we can advance this field? So we need a more physiologically relevant obesity model that clearly distinguishes diet component from the body weight and obesity from the insulin resistance. Because we think that if you have a suitable model, it's very critical for the mechanistic study. And if you have a suitable model that incorporates natural history estimate of the disease progression, that will allow to better understand the mechanism of obesity-linked cancer. We have been interested in this protein, prohibitin, in short PHB, also known as PHB1, for the last several years. This protein was initially identified as an anti-proliferative gene. Even after 25 years of discovery of this protein, we still don't have an attribute of PHB which is responsible for the anti-proliferative function of PHB. So meanwhile, there is a number of other functions had been attributed to this protein. So it's a pleiotropic protein which localizes to different cellular compartment and appears to have compartment-specific functions. Recently, we have discovered that the prohibitin has an important role in adipocyte differentiation. We published in 2012 in International Journal of Obesity. And previously, we have identified a critical phosphorylation site in PHB, that is tyrosine 114. And we have shown that the phosphorylation of PHB at this tyrosine residue is basically downregulate the membrane-associated tyrosine kinase signaling. That include insulin as well as the immune cell type, different immune cell type also. So we wanted to know whether, what are the biological relevance of these two functions of protein, two functions of PHB at the systemic level. And to investigate that, we decided to make two transgenic models. So first expressing the native form of PHB, and second expressing the mutant PHB, having a mutation at the phosphorylation site. To do that, we use AP2 promoter. AP2 promoter is not the best promoter for adipocyte-specific expression. Adiponectin is a much better choice. But we chose AP2 because it was more suitable for what we wanted to do. AP2 is primarily expressed in adipocyte, but it also expressed in macrophage and dendritic cells. So our idea was to simultaneously manipulate PHB function in two different tissues, in two different compartments in a very similar fashion at the same time. So the purpose was to understand the two functions of the PHB, and second goal was, as previously mentioned, the inflammation is the biggest connecting link between these processes. So we wanted to manipulate inflammation and the obesity at the same time. Okay. So let's start with the PHB mice. As expected, both male and female develop obesity, as you can see. We call them mito-obi because they develop obesity as a mitochondrial remodeling adipocyte independent of diet. So they are diet-free obese mice model. Okay, both develop obesity in a very similar fashion, as you can see in relation to their wild-type counterpart. Both develop obesity in similar fashion, starting from the 1 to 2 months of age. On the top, you can make the male, and the green one is the female. Interestingly, despite having comparable obesity, only male develop impaired glucose homeostasis and insulin resistance as determined by the glucose tolerance test and insulin tolerance test. Female mice having comparable obesity is maintaining the glucose homeostasis and insulin sensitivity, very similar to the wild-type mice. So it's a sex differences in the metabolic dysregulation. Okay, this is the structure of the adipose tissue on the top panel and liver on the brown panel. You can see there's the adipocyte hypertrophy in both female and male. But the one difference between these, there is an increase in crown-like structure that is generally formed by the macrophage infiltration. Similarly, in the liver also, you can see fatty accumulation. So the tissue structure are very consistent with the metabolic phenotype of this animal. So we won't go into details because we published this last year in Diabetes. And if anybody interested, you can have a look in the paper. When they age, they develop a sign of NASH. So from the fatty liver, it progresses to NASH. You can see the hepatocyte ballooning, degeneration, as well as immune cell infiltration in the liver. It's a massive infiltration. We confirmed this by immunocytochemistry using the cell type-specific marker antibodies. And from NASH, it progresses to HCC spontaneously. It's around 9 to 12 months of age. As you can see here, it starts with a very small dot, and that is grow in size and is spread throughout the liver. Corresponding histological section is shown in this on the bottom panel. You can see necrosis here, and it's proliferation. Okay, so in the interest of time, I won't go into the details of data, but what we have found that is in the review process currently is the schematic diagram how the disease progresses from obesity to hepatocellular carcinoma. So first, they develop obesity in a sex-neutral manner. The only male get adipose inflammation. This changes the adipokine secretion from the adipose tissue. This leads to hyperinsulinemia in a low ghrelin level. This changes the pro-inflammatory to anti-inflammatory, leads to hepatic steatosis and immune infiltration. This follows the mitochondrial dysregulation, inflammation, and oxidative DNA damage, and finally it progresses spontaneously to hepatocellular carcinoma. In the female, it not only maintains the homeostasis, but it also has a high ghrelin level and high adiponectin level. Adiponectin is known to have a protective effect in many metabolic features, and ghrelin has been emerging as an anti-inflammatory peptide. Okay? And they maintain this throughout the liver, so even with aging, they don't have any problem. So the next question is, what happens to the mutant mice? Okay, mutant mice share the metabolic features of mito-obese mice. So it's a busy slide, but I would like to bring to your attention just on the top two lines, which is basically male PHB, dark one, and the light one is the mutant PHB mice. So they develop obesity in a very similar fashion. They develop impaired glucose homeostasis in a very similar fashion, and they develop insulin resistance in a very similar fashion. So there is no difference between mutant mice and these mice. And female, again, in the same, just like the mito-obese mice, female is well maintained in all features. So this data suggests that the mutation in PHB basically does not affect the adipocyte-specific mitochondrial function of PHB. So what happens, so the question was whether they will develop SCC or not because the features are same. Interestingly, they don't develop SCC. They start to develop lymph node tumor much earlier than the SCC. So the lymph node tumor developed around 6 months of age in these mice, and SCC developed around 9 to 12 months of age. And both SCC and the lymph node tumor develop in insulin resistance and sex-dependent manner. So only male developed tumor, and only those male developed tumor who has insulin resistance. The one interesting thing, relationship between these two tumors is that when the mice develop lymph node tumor, they don't develop SCC. And those mice which don't develop lymph node tumor, they ultimately they develop SCC by 1 year of age. So look like there is some relationship between this, and this may provide some clue how the same set of conditions leads to different types of cancer in human, because obesity is not linked to only one type of cancer. There are 10 different types of cancer that is reported in the literature. So we are currently working on this. So really these two created a lot of opportunity. You can do a lot of things which was not possible before. You can do the mechanistic study of the disease onset. So you can really follow how obesity and obesity-associated abnormalities basically leads to the cancer development and how this will help in the progression. You can do the mechanistic study, the sex differences in SCC. Basically, our model provides a new paradigm in sex differences in SCC, because the earlier, the focus always was on the liver or hepatocyte-specific effect of sex steroid. But what we show that even sex differences in adipocyte and immune cell-specific function is sufficient to lead to the sex differences in SCC incidence. You can do the intervention study because they develop all these features in a very well-defined manner. They start to develop obesity by 3 months of age, insulin resistance by 6 months, and hepatocellular carcinoma by 9 to 12 months of age. So you can do intervention. And most importantly, these mice revealed a sex dimorphic role of PHB, which was not known before. And sex dimorphic role not in one tissue, but in multiple tissues, like adipocyte as well as in immune cells. As I mentioned, the obesity is related to 10 different types of cancer, which is shown in this slide. And coincidentally, the current issue of scientists is basically having a special issue on obesity-linked cancer. If anybody interested, you can have a look in this. There's a very interesting article in this one. If you don't have access, just let me know. I will forward a PDF copy of this magazine. With this, I would like to thank my lab member who really contributed to all these works and the funding support from various funding agencies in Canada. And thank you very much for your attention.